Now look at the graphs. What about the graphs? So what kind of graphs do we get in biology? The same in math, physics, chemistry. As math's going to be a little bit more difficult, but physics and chemistry are basic graphs. Okay, and either you get you have to plot a graph from a given data using this mnemonic ASPL. We'll see what this is afterwards. Or you're given a graph, you're going to be asked questions from the given graph. Two types of questions, the D and E: describe and explain. So the language comes in handy. Now, first of all, we're going to look at the different types of graphs in biology papers are the line graph, the histograms, and the bar graphs. Now, the line graphs can be a single line or they can be multiple lines. So let's have a look at the line graphs. So the line graphs, what are the parts of a line graph? Let's see. Now, this is a simple graph. You can see the x-axis, the y-axis, and there has been some plotting done. Okay, so like for example, these dots that you see are the plotting points, and then you can see a line, right? Okay, so now what are the parts? A, S, P, L. First of all comes the axis labels. So these are the axis labels, the x-axis and the y-axis, right? Now S is for scale would be how you spread your data over the x-axis and the y, and for this thing you need to understand that there'll be a grid that is given in your paper. So your scale should cover more than 50% of the grid. All right, it can go up to 75% and all. It should not go beyond the grid that is given, the graph that is given, and it should not be less than 50%. Right? You're going to lose 1 mark. Okay, so that's for the scale, and then we go on the data points. So these are the plotting points, and how do you plot them? You look at the x-axis and the y-axis. You tally the two, and you can put a small x like that at the data point, or you can put a dot and a circle. So either way, you must be very clear that you do not make it too big or too small, in that it is not visible to the examiner. Okay, and then then comes the line. Now the line, after the data points, you have to join the line, and this is the line. All right, you can use a scale, or you can draw a freehand, join all the points. Okay, so don't worry about that. In biology, not much specifics are given, but still you can just join the graph. Try to be as neat as you can in your graph. And again, yes, please use a pencil for everything that is done on the grid: the labeling, the data points, the scale, and the joining of the line. For all the ASPLs, remember, please use a pencil and be as neat as you can. Don't mess up your graphs. All right. And yeah, so single line graph, single lines, you're going to see a single line like that, right? And this is the x-axis, this is the y-axis, this is the data, or the scale, sorry, and this is the scale. And the axes are fully labeled using the data from the table. So don't miss out any points, and you can see the units are also given. And remember the percentage. Okay, this is the percentage. Remember ASPL, each of this is going to give you 1 mark. Most of the graphs you see will be for 4 marks. So make sure that you complete all the requirements of every graph. Now, what do I do? Okay, D part, describe how to describe a single line graph, right? So first of all, you have to make a general statement of how variables on the x-axis, basically a general statement on how x is affecting the variables on y. The language that you can use is: as x increases, y increases to a certain point, and then if you want to add to it, then you can add if the graph is more than that. Or as x increases, y fluctuates, or as x increases, y decreases. All right, so different language that you can use. So x could be, for example, temperature. As the temperature increases from so and so value, then the rate of the reaction or rate of photosynthesis increases from there to there, or the number of bubbles increase from there to there, right? So yeah, okay, so make a general statement before you start writing it down. Now, how to describe single line graphs. Now, when you see a line, just ignore the second one, sorry, just ignore this one. So if you see a single line like that, now I can see that it's taking different shape as we go along the x-axis. So a part of it is slightly more flatter, part, the middle part is slightly more steep, and then it becomes a little bit more, you might say, flatter. So how do I describe this? Well, split the line into different parts. Yeah, that's it. You describe this. This is A, this is B, and this is C. And so what do we see? This is just for your understanding. You don't have to write it down as A, B, and C in the paper, but just you can split it up into different parts, and you can see this, all of these three are increasing, right? But then you add adjectives next to it. All right, describe each part, and you can quote data from both the axes, and you use adjectives as what? As what? A slow increase, slowly, or gradually, right? So gradually and slowly. And if it's more steeper, so you can use the word sharply or rapidly or more steeply. Any of these words will do. And again, when it slows down, when it flattens out, so you can easily say again that there's a slow increase in the last part. From this point here to this point here, there is a gradual increase, right? So learn to use these words, adjectives. Now, a very important part is quoting data from each part. Now, what do you mean by quoting data? You take 1 point from the x-axis, and then you relate that to the y-axis, and this will give you a total of 1 mark. Remember that. Just quoting from the x-axis, no, it will not give you 1 mark. It has to be from both the axes, okay? So for example, as temperature increases from 0 to 30 degrees Celsius, okay, so that is the x-axis, yeah. For example, rate of respiration increases sharply from this to this. Okay, so that will be from the y-axis. Okay, now this is raw data as you see it from the graph. Now what we can also do is, you want to get more marks, you want to show the examiner that you're much more smarter, then what do? You can do some minor calculations, right? So this is termed as manipulated data. You do some calculations and you put them in your answer. For example, you get it increased from this point to this point, so you minus the 2, yeah, you subtract the 2 values, you get a difference. You get that in the data. This is decreased by or increased by, give a single number. Or you can calculate a mean. You know how to calculate. You can take out a percentage increase or percentage decrease, okay? So yeah, and you can mention the peak value, for example, at for example, whatever the value is, at this value it peaks at this. You can mention the peak. Then you can also state anomalous data. What is an anomalous data? An outlier which does not follow the trend. Sometimes in the paper they will ask you, what is anomalous data? Anomalous data is out of the group. It stands out. It's different from. So for example, you have a certain readings, numbers 22, 21, 28, 22, and in the middle you have 55. This does not follow the rest of the group. So this is something which was an experimental error. We say it doesn't follow the trend. So I'm going to remove it from the data. It's called anomalous data. It doesn't have any significance, okay? Now, what if you have multiple line graphs? If you have multiple line graphs, then you can be, questions will always be about comparison between the two lines. So get ready for that comparison. How do I tackle this? Yeah, so first of all, look for the similarities, then you look for the differences, and you can go on and on like that. Look for, so make general statements for both of them. For example, example would be that both increase and decrease as x increases, both fluctuate over time. They start at the same point, they end at the same point. Anything which you see is a similar concept there, or their peaks have the same height. All right, then will come the differences. The differences would be the starting and the ending points. Yeah, and for example, the starting and the ending points will be the end point of A is in this graph. I just edited it out from another graph, so you can see starting and ending point. So what do I say? The ending point of A, the start of at the same point, yeah, but the end at a different point. A is higher than B. If you have some numbers given here, then you can mention them. So you can say it ended like this. Now compare the steepness of the graph as x increases from 1 to 3. Then you can see B increases from 1 to 4. Yeah, and whereas what do we say about y increases greatly up to all the way up to the number 8. Yeah, changeover point, well, at the beginning is different and they swap places, and you can see there's a changeover point here. A is higher than B in the beginning, but at point X, B, so A is higher here, but in the beginning, at the end it becomes lower, where B is lower, and at the end becomes higher. So you can use this as well. Now the constant phase, at what phase do they become constant? Well, A becomes constant faster than B. You can mention the units, you can compare the peaks. So the peak of enzyme A, the pH is showing at 2, whereas for B it is showing at pH of 10. Each one reaches the peak, calculate the difference, or you can calculate the difference and quote the value as well. You can compare the ranges. For this range, this enzyme pepsin, the range is between 0 to 3, peak being at 2, whereas for this amyl is the optimum is 7, but the range is between 5 to 9. This will also give you another mark. And then rate of photosynthesis, generally we have multiple graphs, you've seen them, multiple line graphs, effect of light intensity and carbon dioxide. So you can see this, and then it'll always be an area where they'll be asking about the limiting factor. So yeah, I don't have time. So this is basically when you want to explain the concept that comes about. Explanation will be that you have to give the reasons why does it go more steeper here when you have low carbon dioxide, when you have high, so there'll be more photosynthesis. So even at the same light intensity, there's going to be more photosynthesis, so the rate will increase as CO2 is the substrate. In photosynthesis, we have to mention the concept of limiting factor. Now for enzymes, for example, the explanation will always have to be about the lock and key mechanism. You have to bring in the words of the enzyme's active site being complementary shape to the substrate, and if it gets denatured, the enzyme, the substrate will no longer fit into it, and hence the reaction becomes zero. So no enzyme substrate complex is formed, and you can go on. All right, so I hope you know this. Now histograms, right? So what are histograms? Histograms are a graph which looks like this. Yeah, so what do we see? Well, we do have an x-axis. Yeah, we have a y-axis, but what's this in the middle? Each of these is termed as a box or a rectangular box, or we can also say columns, right? And so, and if you can draw a line connecting the marks, so you can make a shape like this. This is termed as a bell-shaped graph or a bell curve. Okay, bell-shaped curve or histogram, right? Now what's a histogram? Is similar to a bar chart. We'll do a bar chart in a few minutes, and this is going to be used for quantitative continuous data. If you want to see the terms, you can go on look at in the maths. And x-axis we divide into intervals or rectangles which touch each other. There are no gaps between the boxes, the columns, and the y-axis has to be the data as this is a mathematical term, as whatever the number that is given is as frequencies. They can be percentage as well. Now the plotted frequencies of values for each interval rectangle. Look, so now for example, have a look at this. Now table 2.2, yeah, shows the percentage of men and women in the sample who have anemia. Anemia is lack of pallor. It's a disease because of lack of iron, and there's a deficiency of iron or hemoglobin, and hence a person seems to be pale in color. Now you have to plot a histogram to show the percentage of men and women with anemia in each age. So there are 3 groups, 1, 2, and 3. So they've given a group of these. So there are 3 groups, and the percentage with anemia, men and women. How do I plot this? Now this obviously is the group that you had selected, so this becomes my independent variable, and this will come in the first column. Okay, this is in the first column, but this will come in according to our fix. This will come on the x-axis on the graph. Okay, now the rest of it obviously is the dependent variable, and this will now, according to the mnemonic dry, this values will be plotted on the y-axis. Okay, so men and women, yeah, and we have a certain number of these. Yeah, the percentage, you can't write a percentage here, so this is understood that there's a percentage that goes over there. Age group, I think this was given in the paper, but it should have been given as years here. Okay, so now moving on. Now how do I plot this out? Age group is this. So what do I do? This will come here, and this is the grid. The grid is given. Okay, the grid is given, and this will correspond to the data that is given to you. Now, how do I plot it out? This is a quick look at this. So the age groups will come over here, but mind you, I need to plot boxes. So the boxes have to be, there are 3 boxes, and they have to fill up from here to here. So I can count the number of these and divide them into 3 regions, right? So there will be 3 regions for each of the group, and then for the men and the women, I can further divide each group into 2 so that I can plot the men and the women data separately. Okay, so that will be the x-axis. What about the y-axis? So the y-axis will have to be something to do with the number or the data. Now let's have a look. Okay, so the x-axis, let's come here, the age, the groups, and they're written over here. Yeah, so I've plotted them, and it's coming as more than 50% of the grid that is given. So 1 mark I get for that. Now, so if I want to write down A, S, P, and L could be the columns here. So did I label the axis? Yes. This is the x-axis, and the percentage with anemia. Yes. So I get 1 mark for labeling the x and the y-axis. Okay, good for you. And now if I'm going to write down, make 2 columns for each, I must give a legend on the side here. And have I done that? Yes. So maybe that will come in the last part. You can add 1 mark here for that. And now, so this was for 5 marks. So obviously it's an important graph. And then, yeah, so percentage with anemia, and now we're done with the x and the y axis. Now, yeah, men and women. So the groups have been written down over here. For each group, I'm going to use, in this graph here, in this my PowerPoint, I've used the blue and the red colors. Remember, in your exams, you can't use blue colors. You either given a black pen or a blue or a pencil. So either you use I just want you to use pencils because you're going to make blunders in this graph. So please use pencils only. How do I demarcate the 2 columns? What I can do is I can draw lines on them, and I can show the direction of the lines, and that will give me the 2 columns, the differences in the 2 columns. For men it will be in this direction, and for women, so you can do in any other way if you like. Now, how do I plot the y-axis? Okay, so first of all, yeah, let me just clear this out now. X would be equal width, for each column would be equal width, so I have to count the number of small boxes, right? Same for both of male female, and no gaps in each of the groups or within the male and the female columns, right? So I hope that is understood. Now comes the y-axis. Now the y-axis, so what I do, I'm going to look at the smallest number, and that comes out to be 7, and the highest number is 26. So I can take the data from 0 to all the way to 30. Yeah, so I can take it from 0 to 30. Yeah, and in the middle these 2 values will fall. Now, so this is a lot of space for that. So what I discovered was I can use 2 small boxes for 1 single number, for 1 unit. All right, so the y-axis, 2 small squares is equal to 1 unit. Okay, if you want you can write it down, otherwise you can just plot on the graph. So 0 to 10, 0, 5, 10, 15, 20, 25, 30, and then when you plot the graph, so at the first group for men it was 7, that ends over here, and then for women it was 8.5, and then for the other group it's higher in the males, so this column becomes higher, and in the females it was this, but for the higher age group 85 to 94 the same trend follows. So the male having a higher percentage of anemia and the women having less. So you plot them and you get a graph like this. Remember, there are no gaps in between the columns. Okay, so let's move on. So now you can take up, if you have for each group, right, if you have like male and female, you can draw them on the side like this. All right, remember there are no gaps here, right? So you can draw them side by side, or you can draw them like this as well. Okay, but you have to make sure that you put lines, but there must be some demarcation between the area which the column, part of the column which covers for the male data and for the female data. Okay, yeah, now the bar chart is easy. Bar chart is graphical display of data using bars, or they can be horizontal and vertical using discrete data or discontinuous data, what we call qualitative data, and which you can't, bars need to be of equal width and there will be equal gaps in between. All right, for example, the color of the car, the numbers seen when you plot them, it looks like this. All right, now the difference between the two would be there are no gaps here, there will be gaps here, and there are going to be categories in a bar chart, whereas in histograms you can get number ranges. This is a good word for histogram, ranges. Okay, now another one, for example, have a look at this. So use a respirometer to investigate the rate of respiration in 4 animal species. So animal species are groups, are categories. You can't give them a number except of them being 4 in number. So we give them A, B, C, D. So something which has names to it, so we can put them in a group, right? So these are groups, which we call discrete data, if you want to know the term. Now the table that is given, and I can see this is, it's going to be very easy. So this first column is independent, and knowing the mnemonic fix, yeah, so this should come in my x-axis, yeah, and the rest of it is my dependent variable, and obviously this is going to come in my y-axis, right? Now, so the labeling of the axis is extremely important, and here the labeling is animal species, and here you have the whole line given here. So remember, you want to get full marks, well, you need to put this down on the grid. Now, am I been given a grid? Yes, I've been given a grid. Now, the question demands me to plot a bar chart on the grid to show this of the drop for the 4 animal species. Okay, so the 4 animal species, this will come on my x-axis, keeping in mind fix, and then for the dependent variable, this one, so this will be coming on the y-axis. Okay, but keep in mind this is the whole thing that has to be transferred. Now, because they're saying mean, so there were 3 repeats and they calculated an average, in other words the mean. So I need to add the word mean to my labeling, because that is a requirement of the question. Okay, so this is x and this is y. I'm going to plot 4 of the columns here, and they should be of equal width with equal spacing in between them, and the data that I get is only going to be this and this. So A was 1.5, B was 0.9, C was 2.5, and D was 1.9. Now again, on the y-axis, how do I make a scale? Remember, the scale has to be covering more than 50% but not go beyond the grid. So it has to be between 50 and 100% of the grid. So the lowest number here again, yeah, the lowest number again, let's have a look. The lowest number here is 0.9 and the highest number is 2.5. So I can safely start from a 0.0 number and go all the way to 3.0, right? And this grid is pretty long, so I can use 2 small units for each one of them, and this is what I'll get. All right, so animal species, the columns come on the x-axis, and remember the labeling is extremely important on the y-axis. So this will give you, again, let's have a look at the mnemonic ASPL. A, did I label the axis? Yes. And did I label the groups A, B, C, D, and the whole? Did I mention the units as well? Yes, I did. So the scale, does it cover more than 50%? Yes, so 1 mark for this. And the plotting, obviously the scale, and you plot the columns, and yeah, you get 1 mark for this. And This would be maybe for one, there are no gaps in between the last thing. You don't have to draw the lines. You can just talk about the columns. Yeah, and are there no gaps? If you make no, there has to be, sorry, there have to be gaps. So if it's going to be no gaps, then you're going to lose marks again. Okay, so yeah. I hope you learned something, and thank you for watching, and do leave some information in the comments, any comments that you feel was missing, or if you any other topics that you need to pick up. All right, so watch out for other videos, and again, like, subscribe and share. Thank you so much for watching again.